I'm sitting here reading some of science's classic tales of discovery, the double helix and one of my favorites, the microbe hunters. And they both reveal that whether you're a DNA detective or a tracker of tsetse flies, scientific research requires far more than curiosity. Welcome to Anatomy of a Lesson. On today's program, we'll be looking at the real world of scientific research, how it works, what it costs, and the rules of the game. From organizing science fair projects to keeping aliens at bay, science is often strictly business. The business of science, today on Anatomy of a Lesson. While vigilance is a watchword for all scientific research, there are certain situations where it may mean your very survival. At the U.S. Army's Medical Research Institute of Infectious Diseases at Fort Detrick, Maryland, daily battles rage against the smallest of assassins.
Our primary mission is to develop medical countermeasures to protect our soldiers, our military forces, against biological warfare agents on the battlefield. The other part of our mission is to work with exotic viruses, those which require special containment, which our soldiers might run across.
Back in 1989, one of those exotic viruses came dangerously close to home. As readers of the best-selling book, The Hot Zone, know all too well, when imported monkeys began dying in Reston, Virginia, investigators feared the cause to be Ebola Zaire, an African virus known to kill 80 to 90% of people exposed to it.
We sent a team to Reston and collected additional samples. Our team was equipped with gear that allowed them to work safely, essentially portable BL4 biosafety level 4 gear, complete head covering and respiratory protection and suits that allowed them to work in this facility.
But it was back in the hot zones where the mystery was actually solved. The discovery of Ebola Reston, a new strain lethal only to monkeys, was made by Pillsbury doughboys and girls on a serious mission. The specter of killer viruses on the loose is, for some students, science at its most dramatic and irresistible. Fan letters to the real-life stars of the Hot Zone testified to youthful imagination.
This happened to be a very interesting letter from a young fourth grader, I believe, who was very enthused about the possibility of his eventually working in a place like USAMRIID. And so he wrote Nancy and described to her a spacesuit that he was developing for himself as part of a science project, and it was very interesting, in which he took his sweatsuit and covered it with duct tape and then built a helmet out of a surgical mask and more duct tape, and wore gloves and his rubber rain boots and taped the gloves and the boots to the spacesuit to attempt to simulate working in a BL4 situation.
Ebola can be deadly, but other pathogens can be equally insidious, even if they don't directly affect human beings. The brown citrus aphid, for instance, is more than just another sap-sucking pest. This alien invader is actually a kind of Trojan horse, carrying within its body the Tristeza virus, capable of wholesale destruction of orange groves. To study such foreign threats to American agriculture, a special containment facility operates not far from USAMRIID. Like its biosafety level 4 cousin, this prison for plants makes sure that microorganisms checking in don't check out.
The building itself is built almost like a fort. It's sealed tight, it's under negative air pressure. The water that we use inside goes through steam sterilization. The soil that we use inside goes through steam autoclaves. We have to shower out. Everything is controlled. Nothing alive goes out unless it's inside a special container for processing in the lab somewhere. No live plants, no live insects get back out of here. We bring them in, we work with them in here, they don't go back out.
Research facilities like the ones we have just seen, and those here at the National Institutes of Health, cost tens and sometimes hundreds of millions of dollars. Add to that the cost of staff, maintenance and supplies, and pretty soon even the simplest investigation becomes a big-ticket item. Which begs the question of not where does the buck stop in scientific research, but where does it begin? According to figures provided by the Department of Health and Human Services, national support for research and development in just the health fields alone totaled $31 billion in 1993. That grant money, provided almost entirely by the government and private industry, was awarded to a long list of agencies, research labs, universities, and hospitals. How to get that money and what it can be used for is decided by organizations like the National Science Foundation, an agency with a lot of practice at granting wishes.
A grant is a funny thing that way. It's giving money to somebody to allow them to do something that's very controlled by their creativity. We're enabling their creativity to be expressed. We fund very broad research, anything that helps us understand the unknown, whether it's on Earth, in the universe, inside cells, inside the Earth. Anything that might be of importance scientifically in the future, we want to explore. We also want to help develop the next generation of scientists and engineers. So that's part of our mission, helping to develop human resources for the future of research. We also have to look out for other aspects of the health of the research endeavor. Are there enough laboratories? Are there enough big pieces of equipment like telescopes, ships? We have planes. All of these need support to help scientists and engineers have the capability to do research on things that we haven't even thought of yet.
Of every 3 grant proposals submitted to the National Science Foundation, only 1 is actually funded. The winners pass through a careful grant review process that evaluates a project's worth, its timeline, the principal investigators, and of course, its budget. But even once the cash is in hand, the scrutiny doesn't end. Not only do researchers have to justify expenditures over the course of their grant, but to keep the cash flowing, they have to run the competitive gauntlet once again.
Well, actually, it's a lot like salesmanship. If you're establishing a reputation for a quality product, and quality here is measured not so much by volume, but by creativity and believability, you have to let the consumer know. The consumer in this case are other scientists, and they're a very tough crowd to sell ideas to. They have a high threshold for believability. You have to show them the data. You have to express your ideas clearly. You have to be convincing that you said what you did what you were going to do, that it does work, and it is as important as you said it was back there in your proposal.
Well, the first grant that I wrote was a very small grant as a master's student, and it was to a university funding pool or funding agency. And I guess I was just naive about how I had to explain what I was doing, and I assumed that people would understand it even if I didn't explain it, real clearly. So I guess what I learned was that you have to assume that people are intelligent but not experts in your area at all, and you need to explain very clearly the fundamental reasons why you want to do the research, and then also explain clearly how you're going to collect the data and demonstrate that you are pretty confident you'll be successful.
Success doesn't always come as neatly predicted by the scientific method. Research protocols are often as varied as the studies they describe. But whether the goal is to discover what big teeth can tell about a wolf population, or how chit-chat is linked to a bat's identity, the journey has some common features.
The things that I've had to do to be able to do my research that I think are relatively general are initially conceiving of the idea, and that requires a lot of reading of what other people know and thinking about what people don't know. Writing a grant so that I had the funds to go to Trinidad and do my field work there and bring the bats back, and then supported some of the work I do here. Writing another grant so I have salary while I'm doing it.
After careful collection, measurement, and analysis of valid data, it's usually time to organize the results and reach some conclusions, and not just for intellectual satisfaction.
For a couple reasons. One is to disseminate the information and convey it to your peers, and hopefully also to the public at some level. And also, if you want to write another grant, you have to show that you've been productive with the money that you've had. So that if an agency like the National Science Foundation gives you money to do research, they want to see that you actually did that research and that you actually were able to address the questions that you posed and come to some kind of an answer.
In private industry, research derives most of its support from company profits rather than external grants. And in the case of Biospherix, a company working for years on a non-fattening artificial sweetener called D-tagatose, the payoff is still a sweet daydream away.
At first you think it is something you can do in your garage. You have an idea and you run to the lab and say, hey, that works, and you immediately start fashioning slogans to sell it with. But there's a long way to go. I think the best example is the new product Olestra that Procter & Gamble put out, which is a counterpart to a non-fattening sweetener. It's a non-fattening fat, or fake fat as it's called. Procter & Gamble said it took them 20 years and $200 million to bring that product out. Now, as I say, it's a comparable product. Well, doggone it, we've almost used up to 20 years, but believe me, we haven't used up the 200 million.
When human beings volunteer for scientific research, they're often defined by the animal, often linked to the laboratory. Those guinea pigs, plus mice and rats and other creatures, great and small, have long endured experimental procedures, some of them quite controversial, to advance human understanding. Today there are strict guidelines for the humane treatment of laboratory animals, and indeed for the humane treatment of human beings as well. But it has not always been so. One of the greatest shames of American science is the infamous Tuskegee syphilis study. For 40 years, beginning in 1932, the Public Health Service studied the effects of untreated syphilis on black men in Macon County, Alabama. The lack of any formal procedures and the denial of treatment to the unfortunate participants made this, according to author James Jones, one of the country's most morally reprehensible experiments. The victims were even denied access to penicillin when it was discovered in the 1940s, a drug that might have saved them from syphilis's sentence of death. Today, government protection of human research volunteers is far more enlightened. Federal regulations require informed consent, something missing in Tuskegee, before any experimentation can occur. And only rarely are there exceptions to that rule.
The requirements of informed consent are really key. And investigators have to think about and have to have an awareness of how to give informed consent and how to involve the patient almost as a collaborator, not almost as a collaborator, as a collaborator in the effort. Not so that they tell the patient or convince him to act a certain way or whatever, but that the patient realizes they have to come back, they realize that they may be taking a placebo, they realize that what they are involved in is research and not treatment.
One such treatment recently qualified for a waiver of the government's requirement of informed consent. To save the brain cells of patients with severe head injuries, NIH researchers have found promising results with, of all things, ice water. By cooling the victim, this hypothermia treatment, if started within 6 hours of the trauma, seems to slow the chemical reactions that cause cell death. But since most head injury victims arrive in emergency rooms unconscious, getting informed consent before the window of opportunity closes is often impossible. So the waiver is an important concession in a well-planned study that never forgets the sanctity of human life.
You spend as much time planning your experiments as you do executing the experiments. And that planning stage is really essential, because when you get into the larger studies that are multi-centered and that are millions of dollars, you don't do them more than once. So that the information that comes out has to be solid. And you have to do everything as an investigator to utilize the taxpayers' money appropriately and to protect your human subjects.
Animal lives are also in our hands, and that responsibility has made their role in research one of passionate debate.
Our society is in a quandary with use of animals for many purposes. We use them in biomedical science, certainly for humankind, and many teachers want the opportunity to start precocious kids, to learn something about biology. And I would hate to have gone through school and not have had a goldfish or a bunny or some animal in my classroom to learn something about. I think society fears that we're being harmful to them, when in fact I think we're teaching kids a lot about the care for these animals, responsibility for them, and it's not all about dissection of frogs.
The rats and mice that make up 90% of the animals used in biomedical research are, because of the sheer number of labs using them, outside the realm of government regulation. But the Animal Welfare Act, passed in 1966 and amended in 1985, does protect dogs, cats, rabbits, guinea pigs, hamsters, and non-human primates from inhumane treatment. At the schoolhouse level, the ethical treatment of animals is outlined in a document called Principles and Guidelines for the Use of Animals in Pre-College Education. Published by the Institute for Laboratory Animal Resources, it holds students to almost the same behavioral standards required of professional researchers.
For that occasion, when there's a student that you really want to use an animal in a special way that might harm that animal, we think that the only reason that you should really do that is to train that student as a prospective scientist. And that student should have to go through all of the basic hoops that a scientist does. They should know what's been done on that study, and they should plan a protocol, and they should have people review that protocol who know something about that study, a local veterinarian, a physician in the community. They should have somebody outside the school.
In addition to careful protocols and peer review, the guidelines and principles also specify that no animal be subjected to aversive stimuli in behavior studies, that no animal be given deficient or toxic amounts of nutrients, that all animals should be healthy and free of disease, and that plants or invertebrates with simple nervous systems be substituted for vertebrate animals whenever possible. Developing sensitivities to animal comfort and human safety in the laboratory are important lessons in the education of any scientist. And what better place to develop those attitudes and skills than at some of the nation's premier research centers? At the National Institutes of Health in Bethesda, Maryland, Poster Day has become an annual celebration of scientific talent. From all over the country, hundreds of students from high school to graduate school put on display what they've learned as summer interns in biomedical research. But unlike typical science fairs, Poster Day is no scientific Olympiad. Instead of points and medals, the young researchers get something far more valuable. By talking shop with the scientific community, they're often taking a step into the future.
What I'm doing here is I'm working with a human form of the mad cow disease. It's a spongy form of encephalopathy that occurs in humans. And what I'm doing here is I'm coming up with a way of diagnosing this in humans, because a lot of the symptoms are similar to the symptoms of other diseases.
These are real research projects, we just don't give people busy work to do or that type of thing. These are research projects that will hopefully be incorporated into publications and that type of thing. So it's a real experience. So I think more so than other programs that I had been involved with in the past, it shows you what the nuts and bolts and everyday activities are in scientific research. So it really gives you a very good idea. So I think it helps students be able to decide whether they want to go into research or not.
To hear the students tell it, their experiences at NIH have been a revelation. The rigor and the intensity are far different from any high school experimentation. And yet, they've learned how to succeed in the science business by really trying to make their previous schoolwork count.
Research in high school is maybe one class session or maybe a week at most. This project which I'm working on was started 2 summers ago, and it's still ongoing. We're still getting patients, we're still analyzing data. In high school, there is that tendency to say, this is only assignment, it's only worth 100 points, I'm already got some extra credit, so just don't worry about it, I'll just do the next one. And that's not really valid at this level.
At this level, it's a lot, sometimes people go into a lot more depth, but more than anything, you see the applications of your work at this level. Whereas in high school, I might have done lots of projects on, simply how an ice cube would react to something simple, but never understood why it was important. And that's what NIH has done for me. I've understood that perhaps by studying small basic chemistry, I can go on and understand the basics of a disease, and perhaps one day we can help cure the disease or find a way of alleviating the pain it creates. There's a lot more to science than what it is on the surface, but it is all based on the fundamentals we learn early in school.
Those fundamentals of research can perhaps best be learned during a research practicum. This scheduled course, offered almost exclusively in dedicated high schools, allows students to focus on a project for an entire year and often beyond.
Well, first of all, they do at least a year-long project. Some of them actually start as maybe sophomores or juniors in their projects. And what you saw the students doing today is they go out in the field and collect water samples, and they come back and do their little droppings. And but they have to do it every other week for a whole year, and they have to put together a 5-chapter paper on the data they collect. They have to analyze it statistically, which is very different from, it's one thing to have a lab and do statistics on it in a classroom, as opposed to have a lab and have to figure out, what do I do with this data now? Where is it going? Because everybody's doing something different. They also have to do a symposium, and symposium is different because at the end of the year we get all our RP students, and it's not a competition. It's an exchange of ideas, almost like a real scientific conference.
Most research practicum courses also include the study of different research methodologies, the collection and recording of data, communication skills, both oral and written, how to choose a research topic and then submit a formal research proposal, and of particular importance, the ethical considerations of research. Many RP students spend half their school days off-site at research labs with science mentors. And some that are particularly ambitious, as we know, give up their summers as well.
So the anatomy of research is quite complex. It is equal parts investigation, budget, curiosity, and a measure of luck. And through it all, an awareness that the ethics have to be as good as the science. Thanks for joining us today on Anatomy of a Lesson.
Mm-hmm.